my name is Sidney Pontes-Filho. This is the trial lecture for activity defense, and it's titled Comparing Learning Rules in Biological Neurons and in Artificial Spiking Neurons. Since we are going to compare learning rules in biological neurons and is to see how the system works. So it's our brain. This is the system where it consists of many biological neurons and other cells too. And humans, we have five senses that are sight, hearing, smell, touch, and taste. And all of these excite some sensory neurons that are going to send spikes. Those spikes are pulses sent from these neurons. These are like binary strings, like 0 and 1s in continuous time. When there is a spike, it's a 1. When there is none, it's a 0. And it goes in a flow through the other cells. So it passes from a neuron to another and get this connection. And it's processed with other neurons inside the brain. And then it's sent from our sensory, from our sensors to our muscles. So my voice is also a series of muscles that are projecting it. So what I'm seeing, hearing, and everything, and processing inside my brain, I can activate my muscles and perform my action. And to have an idea, since our brain is a learning machine, for example, our eyes, the reaction for some stimuli is like 200 milliseconds, but for our hearing, it's much faster. And you can also speed the reaction time. For example, some game, like a first-person shooter game, or some goalkeeper in the football, like they reduce the actual reaction time by training, and fortifying pathway from the vision to the hands or muscles. But the normal person has 200 milliseconds. So yes, and from the brain, we have artificial, sorry, because it's not just him. Yes, and we try to make this brain, like we try to imitate it. And the closest that we have is a spiking neural network. So we try to model the brain in a simple way. And we also have, as a sensory input, like spikes. We can give spikes to this neural network. And it's also being processed by other neurons, and also send output or activate in some way to control a robot or to identify a class. For example, it's a dog or a cat. And also it's going to be spikes. Our muscles also react to spikes as well. And the components of a spiking neural network are the spiking neurons. And you can see them animated in the right side. And yes, we have spikes coming to the neuron. It's going to have a neuron state that's going to be activated or increased. And when it reaches a certain threshold, it's going to output another spike to the next neuron. And this is how the neuron works. I'm going to give more details to it later. But in this lecture, I'm going to be going from, since I've been comparing biological and artificial, I'm going to be switching from biological to artificial, biological to artificial. First, we saw the brain, the biological part, and then the spiking neural network. This was the system. Now the unit of the system that I'm going to explain, and the unit of the brain is the biological neuron. So these are the components of the biological neuron. The dendrite is like, it acts like an input. It receives signal from other cells. These other cells are presynaptic neuron. We have the soma, the cell body. Our cells have always a nucleus. And to give kind of when it gets excited, this neuron, it sends action potential through the axon and then reaches the axon terminal. And from there, it sends to the postsynaptic neuron. That is the neuron that's going to receive this information. And also it can be connected to a muscle, for example, to activate the muscle. So this is the neuron. This is the postsynaptic neuron that receives the information from the presynaptic neuron. We have presynaptic neuron and postsynaptic neuron. A synapse is a connection that has a direction. And this direction is from the presynaptic neuron to the postsynaptic neuron. And that blue one is the postsynaptic one. Okay? And for the one that we have in the middle, we have the presynaptic one that gives information or spikes, for example, action potential to the next one. And how synapse work. And is explanation how kind of a zoom in to a synapse. the presynaptic neuron has some neurotransmitters. Normally, it's a glutamate. It has some vesicles that hold those neurotransmitters, the synaptic vesicle. And when the cell is excited, it's going to release those neurotransmitters to the postsynaptic neuron. And this happens when they're excited. It happens this docking. It's like lots of proteins. Our cells are protein machines. So docking, fusion, trying to open the presynaptic terminal, and then releasing the neurotransmitters that the postsynaptic neuron is going to have receptors that are going to get those neurotransmitters, and then excite or inhibit that postsynaptic neuron. This is a zoom in, and to understand a bit how this process happens in the neuron, I'm going to explain the artificial spiking neuron. It gives kind of a very abstract model of how this works. And this explanation of this model, leaky integrated fire model. Here is our spiking neuron, the left side. It receives spikes, or it receives spikes and output and really sends the spikes as well. But how those spikes are generated by a spiking neuron. So on the left side, on the right side, we have input spikes coming to the spiking neuron. This spiking neuron has a neuron state that is normally in the resting state, that is touching the T or the horizontal axis. But it arrives one spike to this spiking neuron. And then, regarding to a weight of that connection, a W, it increases neuron state to a certain amount. And when there is no spike, no activation, this neuron state tries to decay exponentially to the resting state. But when more spikes comes to this neuron, it compensates this exponential decay, and then this neuron state increases, or maybe can also decrease if these are all excitatory spikes, but can be inhibitory spikes that reduces the neuron state. But when this neuron state reaches a certain value, the threshold, it outputs a spike, and then it resets the neuron state. This is how output spike is generated using this model, leaky integrated fire, and it's kind of very similar, just a very abstract way of how our biological neurons work. And in the top is the formula. We have delta V is the change in the membrane potential in a certain time step, that is delta T. And we have the inputs, that is this multiplication of the weight with the input. Normally speaking, this is 0 and 1. The input would be just only of these two numbers. The weight defines the change of the neuron state. We sum with all the connections that this neuron have. And then the minus delta T, lambda decay V is the exponential decay. Delta T is the time step. The lambda decay is the decay factor. And V is the voltage or the membrane potential of that neuron. And this is how a spiking neural network basically is. So as I explained before, we have excitatory and inhibitory neurons. And how do they work and how they affect the postsynaptic neuron? So as we have excitatory neuron that are represented by green color, inhibitory neuron represented with red color, and the postsynaptic neuron represented with blue. We can see that excitatory neuron fires first. You can see in the horizontal axis of the excitatory neuron. It creates a bump, like it activates a bit the postsynaptic neuron, but does not produce a spike. But then there is a second and a third excitatory neuron that goes very close together, that makes these the postsynaptic neuron be excited enough or having its neuron state, maybe potential, to produce an output spike. And then it resets and everything, and then it comes to, and then it reaches the resting state or the resting membrane potential, and then it comes a inhibitory spike, and then it reduces these resting state to below that, and then we have a second inhibitory spike. You can see the reduction in the postsynaptic neuron, but then it gets two excitatory spikes in sequence. It could produce an output spike or a postsynaptic neuron to have action potential, but does not produce because it does not reach the threshold. And this is how the excitatory and inhibitory neurons affect biological neuron. And also we try to do with artificial spiking neurons too. And how the biological neuron kind of work, kind of to understand as well how the learning rules is going to be also working. So we need to understand kind of the mechanism of biological neuron. And this is how the neuron maintains its potential that is minus 70 millivolts. We have kind of a voltmeter there measuring the difference between the extracellular fluid and the internal part of the cell. So how to maintain these minus 70 millivolts? The neuron needs to expend energy. And what we have in the outside part of the neuron, we have more sodium ions than potassium ions. It's mostly sodium ions, but in the internal part of the cell is mostly potassium ions. And we have some potassium channel and sodium channels. They are triggered by voltage of the membrane potential, or also by some neurotransmitters or chemicals. And we have like the sodium and potassium pump that spends ATP energy to maintain this level, this difference of membrane potential, and also the sodium ions outside and potassium inside. And this is how action potential work happen in the biological neuron. So how the spike is performed by the neuron. So we have the minus 70 millivolts as the resting state or the resting membrane potential, and the threshold is minus 55 millivolts. And when we have some stimulus, maybe it's not going to reach the threshold. It's shown in the failed initiation, but maybe some initiations trigger and pass the threshold. So we have a phase that is called depolarization. It's mostly the sodium gates are going to be open, allowing sodium from outside to enter the cell. And then we have a peak that is 40 millivolts. And then after that, when it reaches these 20 millivolts, the sodium channel is going to open, allowing potassium ions to go outside of the cell. And then we have the repolarization that goes to the resting state. It overshoots these opening of channels and causes the hyperpolarization. And then after a while, it goes like the pump working everything to maintain into the resting state of the resting membrane potential that is around minus 70 millivolts. So yes, there is this work with the sodium channels and potassium channels to trigger these spikes and communicate to the other neurons as well. Okay, and here is how the effect of actual potential to the postsynaptic neuron. How each spike is kind of perceived inside to the postsynaptic neuron, like the membrane. We have first the twitch, is just one spike. This is going to be important for some learning rules that we are going to learn later. And here we have a twitch, just one spike. When we have three spikes kind of in a considerable space between them, we have the summation. And we have these that's called the rough tetanus. Tetanus is something important to learning in biological neurons. That's why I'm explaining it. So when we have kind of certain frequency, a fine rate of spikes, it has all these rough tetanus, that is the membrane potential of the postsynaptic neuron increasing, but with a certain roughness because the decay is also kicking. And we have this small tetanus too, that the fine rate is high, that just it goes smoothly without allowing the neuron to try to go to the resting potential. And this is called smooth tetanus. Okay, so let's talk about neuroplasticity. So for comparing learning rules, I thought how these two neurons work, the biological neuron and the artificial spiking neuron, and how those two neurons are going to learn, how they are going to modify to learn something. And the way that they learn is, of course, using neuroplasticity. And neuroplasticity is the change of communication between the neurons over time. Maybe a neuron is going to receive a certain input, a certain stimulus, and output a certain quantity of spikes, but maybe the same stimulus is going to change it, the amount that this neuron is going to send to the postsynaptic neurons that it's connected to. And this change depends on past activity of the neurons. And we are going to see that in the next slides. And we have two differences in biological neurons And in artificial neurons, normally, yes, what we're going to explain here, in the changes in the biological neurons, it changes the number of receptors of the postsynaptic neuron, the number of neurotransmitters in the presynaptic neuron, or maybe the changes in synapses. You can change the structure or change the number of synapses between one neuron to another. Since artificial or spike neural network is a simplification, the only change in the connection weight and in the threshold of that spike neuron. So yes, Hebbian plasticity. Donald Hebb came to a theory about how neurons learn, and I put this quote about him. I'm going to read it. So yes, when Donald Hebb say, when an axon of cell A is near enough to excite a cell B and repeatedly or persistently takes part in firing it, some growth process or metabolic change takes place in one or both cells, such that A's efficiency as one of the cells firing B is increased, or shortly saying, cells that fire together wire together. I think some of you have heard about it. So yes, when two neurons, presynaptic neuron and postsynaptic neuron, fire close, and maybe depending on how they work together, they are going to increase that connection. The synaptic strength is going to increase. And how these Hebbian plasticity work? We have this long-term potentiation, that is LTP. It was good that I explained biological neurons first, because we're going to see these sodium ion, potassium ions, also magnesium ions and calcium ions. Okay, and we have two types of receptors. They are AMPA receptors and NMDA receptors. And we have some red circles that you see in the top of the videos that are the glutamate neurotransmitters. And the AMPA receptors, when it has some glutamate close or like attached to it, it's going to allow potassium from outside, from inside of the cell to go out, and sodium to inside, increasing the excitability or the membrane potential inside the cell. And the NMDA receptor does the same thing. It allows sodium to enter the cell and potassium to go out, but it's blocked by a magnesium ion. But when the magnesium ion is not there, blocking the NMDA receptor, it allows as well to enter calcium. And calcium is what triggers to add more AMPA receptors and allowing the postsynaptic neuron to be more sensitive to the stimulus from the presynaptic neuron. And I'm going to show the video. Here's the glutamate coming to the receptors, and you see that attached and allows these receptors to send sodium inside the cell and potassium to go out. But you can see that NMDA didn't change, didn't send anything because it was blocked by a magnesium ion. And you can see the change in the membrane potential. It was just a small change. But during tetanus that I explained about the firing rate, like high firing rate, the postsynaptic neuron maybe triggers that. So you can see the glutamate coming again to these two receptors, AMPA and NMDA. But since there was some membrane potential inside the cell, because it was activated before, now the magnesium is not going to be easily attached to the NMDA receptor, and allows sodium and calcium ions to enter the cell and potassium to go out. Since with depending on the concentration of calcium ions, it triggers the addition of AMPA receptors. You can see that the third video, we have additional a second AMPA receptor, and this facilitates the membrane potential to increase and facilitating this cell to create action potential or to fire. So you can see in the video again, glutamate coming to these three receptors, and now the membrane potential is much more excited, easily excited. The membrane potential is more like increases much easily because more sodium enters the cell. So yes, this is kind of the mechanism of LTTP. And we have three types of LTTP expressions that can happen in a biological neuron. We have on the left side, we have two AMPA receptors, two NMDA receptors. We have the kind of four vesicles of neurotransmitters. It happens the depolarization that sends the neurotransmitters to the postsynaptic neuron. It enters sodium and calcium ions. And the third one, the induction, the quantity of calcium ions triggers this CMA kinase that pushes some changes in that synapse. And it can happen expressed in three ways. In the presynaptic neuron, increasing the amount of neurotransmitters to be released. It can happen both, like creating new synapses, or in the postsynaptic neuron, adding more AMPA receptors and increasing their response. So yes, the discovery of long-term potentiation happened in Oslo in 1966, and it was done by Terje Lomo when he was working in the lab of Per Andersen. And he was working in some experiments, some neurophysiological experiments on anesthetized rabbits to explore the role of the hippocampus in the short-term memory. And then he discovered that. We have the picture of LTP, long-term potentiation, more AMPA receptors being there, more calcium inside the postsynaptic neuron. And we just not have long-term potentiation. Our cells not just get more connected, it also kind of can lose some of the strength in the connection, in the synapse. So we have LTP from the left to the middle figures. You can see the addition of AMPA receptors, but in case that cell is not excited frequently, maybe the quantity, the concentration of calcium ions in the postsynaptic neuron can reduce, and maybe the AMPA receptor that was maintaining the strength of that connection can be removed in that synapse. So reducing the strength of that connection between these two neurons. And you can see the AMPA receptor being break off from the terminal of the dendrite. And we have in spiking neural networks and also biological neural networks, we have this type of plasticity that's called spike time dependent plasticity, or STDP. STDP is a kind of a tail version of the Hebbian plasticity, neurons of cells that fire together, wire together. And long-term potentiation and long-term depression, LTP and LTD, can happen because of STDP. And STDP is happens because of the spike time of the presynaptic neuron and postsynaptic neuron. It defines the weight change or the synaptic strength of that synapse or that connection. So yes, it's a difference between this spike time of the presynaptic neuron with the spike time of the postsynaptic neuron. And you can see the formula there in the horizontal axis. It takes the time in the presynaptic neuron minus the time in the postsynaptic neuron. And we have that connection is a direction from pre to post. Pre is J and post is I. And you can see in this example, we have pre before post. And because it follows this direction, it follows the direction from pre to post. It happens the spike first in the presynaptic neuron, and then it happens a spike in the postsynaptic neuron. Maybe there is a causality that the presynaptic neuron maybe fired that postsynaptic neuron. And because of this relationship, in this case, it's going to increase the synaptic strength of the connection between these two neurons. And we can see the delta W, that is the weight, the change in that weight is going to increase when pre comes before post. But if the opposite happens, if the post fire first and then comes the pre, there is no causality between that because the connection, the direction of the connection is from pre to post. So there is no causality, and then this weight is going to be decreased or depressed. And you can see in the positive side of the difference of spikes of these two neurons. And we have STDP in biological neurons, and we have several types of them that was verified, most of them from neurons in rats. And we have Hebbian. In these plots, the horizontal axis is shift because it's not pre minus post times spike. It's going to be post minus pre. That's why it's going to change. But it's going to be okay in the horizontal axis. You can see Hebbian, the first column. The first column is related to Hebbian learning. That's neurons that fire together, wire together, or when there is a causality, it's going to increase the weight or the synaptic strength, and when there is no causality, it's going to reduce. We have the type of neurons that we have Hebbian that was checked was FS striatum, NOS interneurons striatum, fusiform dorsal cochlea, pyramid neocortex, and slate etorhinol. And you can see the different colors in the graph. They are from like indicating the difference between the presynaptic neuron and the postsynaptic neuron. The blue ones are from excitatory to excitatory. The green ones are from excitatory to inhibitory neurons. The purple one is from excitatory to polygenic interneurons, a type of neuron that I'm not going to explain. It's just interneuron. And the red ones are from inhibitory to excitatory neurons. And these are, the first column is from Hebbian, and we have anti-Hebbian that does the opposite. That so neurons that fire together, wire together, this is the opposite. Neurons that fire together do not wire together. So you can see when there is a causality, it reduces the synaptic strength of that connection. But when there is no causality, when post comes first and then pre, and then it increases that connection. The idea of anti-Hebbian is to make the presynaptic neuron not firing with a good high frequency. It needs to maintain inactive for a while. This is kind of the idea of anti-Hebbian. And we have also all LTD. It's always a depression, long-term depression. And you can see like neurons that fire together, that the time of that spike is, the difference is equals to 0. There is not much difference. It always reduces. So neurons that fire together do not fire together. For this type of neurons that are the FS neocortex or the shore with high excitability striatum, the cartwheel dorsal cochlea nucleus, and the spiny stellate neocortex. So yes, we have SDP in biologic neurons, and it was verified. And we have other types in artificial neurons, artificial spiking neurons. And we have the type that is asymmetric. And we have, we can make all these plots with these formulas, like with exponential. For the asymmetric Hebbian, we have the delta W, the change of the weight in a certain time step, or not certain time step, but difference in the pre and post. So the spike difference in time, that is delta TR. And when delta TR is greater than 0, or it has it needs to increase the potential of the membrane potential, not potential, the synaptic strength. So we have that formula that is A plus with the exponential of minus delta T R, that's the difference in the two spikes, and this tau plus, that is how this curve is going to be, how it can be much closer, much smaller, this curvature, or much bigger, going much further in the delta TR. And yes, A plus is related to the height of that curve in the positive area when the delta T is greater than 0, and the T plus is just related to the curvature. And when it's negative, delta TR is minus or is less than 0. We have just the opposite from that formula. And when it's 0, the delta TR is equals to 0. No change in that synapse. And for asymmetric anti-Hebbian is basically switching and inversing the signals. And then we have that function of that graph. So yes, to have symmetric STDP in artificial spiking neurons, instead of using exponentials in the positive side, the negative side, we have the difference of Gaussians. We get two Gaussians. For the positive side, we have a Gaussian with a smaller standard deviation, and we subtract with a Gaussian with a higher standard deviation. Here in the bottom, we have the difference of Gaussian function. We have the first Gaussian with we define a sigma plus that is related to the width of that normal distribution minus a Gaussian function with a standard deviation that is equals to the sigma minus. That is going to have a bit larger. So when we make this difference, we have that type of function in what you see in the symmetric Hebbian. So the same for when the Gaussian difference of Gaussian of delta TR is greater than 0, we just apply the difference of Gaussian, multiply with the A, the A plus and the A minus just defines the height of the function. So you can play around how these curves are going to look like. And the symmetric Hebbian is just the opposite. You just change, inverse the signal. And for artificial spiking neurons or a spiking neural network, we have all the ways of training them. We have STP learning, and we have Pro, that is unsupervised local learning. There is no supervision when using this type of learning. And the con is they have deep accuracy and can better used in shallow networks. And we also have a stochastic STP, or artificial spiking neurons, that instead of defining the change of that connection, the weight of that connection, it has a probability to change the weight of that connection to a lower value, to a higher value, or to a lower value to a high value to a lower value. So in the first, we have when the time difference in time is smaller, like Hebbian depression, like there is no causality, it causes the Hebbian depression, like the probability of changing to high to low happens. And there is a Hebbian potentiation when there is a causality area that has a high probability of changing from low to high a weight or connection. There is kind of a dead zone. There is no probability when the time difference of the two spikes are in that range. But if it's the difference is too large, there is an anti-Hebbian depression. Just the probability of reducing that connection. And we have as well two types that is mostly using backpropagation of errors. That is training artificial neural network, how artificial neural network are normally trained with in deep learning. That takes the error and backpropagates to adjust the weights. We can train normally artificial neural network with backpropagation and send the weights that it was learned to a spiking neural network. It has some pros, that is take the advantage of the standard artificial neural network training, but the pros is that incurs some higher inference latency. I also not mention the pros of the stochastic STP, that is high memory efficiency, STP-based supervised stochastic training, and it takes the cons of limited accuracy, but does not have the con of the shallow networks as the normal STP. And then we have backpropagation. We can apply spiking neural networks with backpropagation, but it needs to have some work to do it because a spiking neuron is not differentiable. And then the pros is competitive accuracy and lower inference latency, but there is the higher training effort because the spiking neuron is not differentiable. There is some workaround to work with spiking networks for using backpropagation. Okay, and what is backpropagation of errors? So yes, we have adjustment of the weights of each connection of the neurons using the error in the prediction of the artificial neural network. So we give to this artificial neural network a data, and we want from this data an output that we desire. It gives a prediction, and we have an output that we desire. We compare them, we calculate an error, and that error is sent back through this neural network to adjust the weights of each connection. And this is how artificial neural networks are trained normally and only in the deep learning field. And we are going to compare here backpropagation of errors and Hebbian learning. We have this MNIST dataset that is these are dataset of handwritten digits used for image classification. And we have the top weights with backpropagation. You can see that these are the neurons responsible for 0, 1, 2, 3, 4, and so on, and weights with Hebbian-like learning. You can see the difference of these two methods for training artificial neuron, or spike neural network. You can see that backpropagation is a much noisy representation of a number. And while with Hebbian-like learning, it kind of gets the structure of the numbers. I hypothesize that the Hebbian learning kind of takes a snapshot of the input that comes to the neuron and takes that information. And with backpropagation, it's just a statistical formula to try to make it learn. It forces too much, but for example, the number that is related to number 2, that is in the top row, in the middle, you can see that it is a high weight in the right side. You can see right here, I'm going to just show it. In this location right here, there is a high weight. But are those, these should be firing, like facilitate the firing of that neuron when activation comes in that region? But are those area more responsible to say, oh yes, if there is some pixels activated there, it's going to be certainly a 2? I don't think so. It's not correlated to be a number 2 just because there are some pixels, white pixels over there. So yes, backpropagation has this deficiency of taking kind of the statistical part of the data set and kind of overusing it. And we have supervised learning with SDP for the MD's data set of those handwritten digits. We have input given to a layer of excitatory neurons, and it kind of gets the one that activated the most. You can see that these excitatory neurons, these are like the weights related to the input. You can see that with Hebbian or SDP, it gets kind of the structure of the numbers. It kind of saves each number kind of in the layer of excitatory neurons. And we have another layer that is inhibitory neurons that, for example, here you have a number 5 firing. And that excitatory neuron is going to inhibit all the rest of the excitatory neurons, just to maintain that neuron the most activated in this layer of excitatory neurons. And it's kind of a type of winners take all. Kind of this one is going to have the high activity of all these excitatory neurons, and maybe these are going to be the number that's related to the input. And let's see how it works. Here is the 2D receptive fields. These are the weights that were produced by these excitatory neurons. We can see kind of the numbers where the white region, like where the high activity that happens where the number is, they haven't took where they are located. So when there is a 9, it's going to check the weight and the input. It's going to increase the activity of that neuron. And these are all the 2D receptive fields. When we increase the number of excitatory neurons, that is the graph in the middle, we have the test accuracy increasing up to 95% accuracy. And when we add more and more training samples, that is the one on the right side, we have increase in training accuracy, but it reaches a plateau. We have also a neuroevolution of adaptive neural network. We can also train using evolutionary algorithm. This also happens to our brain in biological neural network with natural evolution. Our brains evolved to do what it does. And this evolution is the outer loop of this learning process, like passing from generation to generation. And we also have an inner loop of learning that happens inside the neural network, inside our brains, or the brain of a robot, for example. So here we have a population of neural networks with different topologies and different weights between the connections. And we test them in a robot performing a task. This network is going to be adaptive, so it's going to have a learning process. That's why we have an inner loop of adaptation to perform the task. And it's going to have also a dynamic environment that needs to outcome that. And we're going to have a selection of the best neural networks that perform that task that the robot is performing. And we select some parents, some individuals to be to change, perform some variation, and then produce a new generation of neural networks that's going to be tested again in this outer loop, and also having all of them the inner loop of learning or adaptation. And this is kind of neuroevolution, our evolving neural network with adaptive neural networks. And here is the idea of looking inside of neuroevolution with adaptive neural networks. We have node genes that defines where is each node. We have input nodes, we have hidden nodes and output nodes. And we also have the explanation of if it's with asymmetric Hebbian or if it is with symmetric Hebbian. These params is related to the height of the SDP that they have. They have a bias that is kind of a noise that always activates the membrane potential to increase. And it defines as well if it's inhibitory or excitatory. These red, the number 4, node number 4 is inhibitory, that's why it's in red. When it's excitatory, like node 3, it's blue. And we have the connections as well. The connection is just defining where, from the destination of a neuron, the presynaptic neuron and the postsynaptic neuron. For example, from 2 to 3, or the first connection gene is from 0 to 3. We have that connection there from 0 to 3. It's enabled. Novation number is related to the algorithm. We have connection from 1 to 3. It's disabled. So we have these enable disabling the connection genes as well. Yeah, and also genes as well have some disabled genes that do not be shown in our phenotype in our body. So yes, this is looking inside of the evolution of adaptive neural networks. And here is a summary of the lecture. As I mentioned, we went to like biological neurons and then spiking neurons, showing the counterpart, the way that we try to understand the biological neurons and the biological neural network and try to model it. So the first one, we talk about the brain, and then we talk about the spiking neural network. Then we talk about the unit. The unit of the brain was the biological neuron, and then we talk about the unity in the spiking neural network, that is the spiking neuron. And we talk about some learning rules in these two networks, in these two systems. In the biological one, we have long-term potentiation, long-term depression, and also the biological SDP, that can have a variety of SDPs for biological neurons. And we also saw the learning using artificial SDP, the formula of them to make those SDP, particularly for artificial spiking neurons, backpropagation, and also their evolution. There are much more learning rules for biological neurons and artificial neurons that I could not cover in this lecture. But yes, thank you very much for your attention.